Hi everyone, thank you very much for the opportunity to speak at this meeting of the Centre for Tropical Health and Emerging Diseases. I'm very grateful to be here to talk to you. My name's Richard Bradbury, I'm a member of the Centre and I'm based in Townsville in North Queensland. I'm sorry that I couldn't be present face-to-face with you today. Unfortunately, I'm not able to make it, but here is a recording of a little bit of information about some of the research which I've been doing involving metabarcoding and pathogen discovery in the context of parasites, and particularly nematodes, which of course are roundworms. Now, if you did some parasitology as an undergraduate degree, you might be familiar with these particular roundworms here. These are the ones which I kind of learnt about when I was an undergrad. And I came out really just knowing about them. When I was an undergrad, there were only two hookworms that infected humans, Nematodes americanus and Ankylostomum duodenale. But over the past 20 years or so, it's become clear to us that Ankylostomum solanicum is a third hookworm of humans. In fact, it's the second most common hookworm of humans in the Asia-Pacific region and ranges in prevalence for anything from 5 to 50%, depending on which part of Asia or the Pacific you're in. And it's a zoonosis from dogs and cats, so it's really important because that has implications for public health control. But what about other things? Here's something which just came across my desk a few years ago. This was a hookworm that was pulled out of an adult male in Perth in Western Australia. Of course, hookworm is not endemic in Perth, and he did have some recent travel to Bali, so maybe he'd acquired it there. And if you looked at it closely, and we did most of this by morphology, you'd say, well, it's got teeth, so it must be Ankylostomum duodenale or solanicum. But the problem is it's got 3 teeth, and they're not quite right to be any of the common hookworms. So I knew what this was. It's Ankylostomum caninum, the dog hookworm, and that was confirmed by sequencing of the COX-1 and ITS genes. So Ankylostomum caninum dog hookworm in a human. Now, this is not actually uncommon in dogs. It's almost 50% of dogs in Australia, certainly in the tropical and temperate regions, carry this hookworm. But in humans, there have only been a small number of cases. Now, it doesn't reach maturity, it doesn't produce eggs, so whether you could really say it's a true human hookworm or not, but I'm going to add it to our list of hookworms. So now we have Nematodes americanus, Ankylostomum duodenale, Ankylostomum solanicum, and Ankylostomum caninum as possible hookworms that can infect humans. So that must be it, right? I mean, we've already got 4 of them. I mean, hookworm's been known about, one of the first parasites to really be known about. So surely there can't be new things to be discovered. Well, some years ago, there were 2 researchers from the Czech Republic. They were primate researchers, and they were doing work on gorillas in the Central African Republic. And they came back with hookworm infections. Now, these 2 people, and I actually know one of them, were very good at parasitology. And they purged their own hookworms, and they identified that these were not your normal hookworms that you'd expect to see in a human. This is what it looked like. This is Nematodes gorillae, first described in 1965 in a Japanese paper. And it's a hookworm of gorillas and chimpanzees, but these were the first confirmed human infections. My colleagues from the Czech Republic went on to find a small number of human infections in villages nearby, where villagers were going out and into the forest areas where the gorillas were and getting infected with Nematodes gorillae. Now, I've had a wonderful PhD student who's just finishing up right now, and that's Huan Zhao. And she's been doing a lot of work with metabarcoding in nematodes. And one of the things which we did was we got a lot of samples from Gabon, rainforest areas of Gabon in West Africa. And it was really fascinating when we started looking at this, because 14 out of 227 samples had hookworm eggs in them. So hookworm was not a common parasite there. But 4 of those were Nematodes gorillae, not Nematodes americanus. And there was another one which had an Ankylostomum species that was phylogenetically between Ankylostomum duodenale and Ankylostomum caninum. We were able to do all this just by doing a single conventional PCR for a region of COX-1 and running it through Illumina sequencing. And we have to use Illumina because often there's more than one species or genotype of the particular worms in these humans. So we've shown now that Nematodes gorillae is not only found in the Central African Republic, but is found some distance away in the coast of Central West Africa. And I'd suspect that as we start looking further, if I was able to get funding to look at hookworm throughout Africa, we might find that Nematodes gorillae is really quite common in some of these forest areas of Africa amongst humans, and that there's a now 5th human hookworm that's infecting humans. And let's not forget that there's another unique species of Ankylostoma where we saw eggs being passed, by the way. All of these were eggs being passed, that we don't know what it is, and it's found in West Africa. So although hookworm's been known about for hundreds of years, there are still new things to be discovered in that area of parasitology. Another worm which has been around for hundreds of years is whipworm. Those of you who did some parasitology in your undergrad may remember Trichuris trichura, the human whipworm. Now, a colleague of mine, Jenny Kaiser from Swiss TPH, was doing surveys. What she was actually doing was doing efficacy of dual albendazole ivermectin treatment for whipworm in Africa, and she found that there was a subgroup of people who didn't respond well. Their whipworms did not respond to dual albendazole ivermectin therapy, which most of the other ones did. So to investigate this further, they started doing metabarcoding, and here's the haplotypes which they came across. Now, there's a tiny little purple dot that's out on the end of that N equals 42 there. That's Trichuris suis, the pig whipworm. And you'll see that there are clearly two populations of whipworms here. And these are samples, this was recently published in Nature Communications, samples taken from Cote d'Ivoire in West Africa, Tanzania and Uganda in East Africa, and Laos in Southeast Asia. And in all of those regions, there were two types of Trichuris in humans, and significant numbers of them, up to 20% of the Trichuris were this other haplotype. And that is a new species of Trichuris, which has been named Trichuris incognita. Here's where it sits in a phylogenetic tree based on whole genome sequencing, and you'll see it's actually quite distant from Trichuris trichura, the traditional human whipworm. In fact, it's closer to Trichuris suis than it is to Trichuris trichura. So there's a new species of Trichuris out there. When we looked at our Gabon samples, we found that we had a bimodal distribution in size. We had these normal-looking Trichuris and these very big Trichuris eggs. And although it's not yet published, the actual ICZN description of Trichuris incognita describes the eggs as being bigger. So we think that's what we're seeing here. And we're in the process right now of doing ITS2 sequencing on all of those samples, because we believe that we've got Trichuris incognita in Gabon as well. Let's now move to Papua New Guinea. Now, Papua New Guinea has always been really interesting when it comes to Strongyloides, because it's got Strongyloides stercoralis like virtually everywhere else in the tropical world and in the subtropics and even in some temperate countries. But it always had this other Strongyloides species, this egg-producing Strongyloides species, which was called Strongyloides philoborni subspecies kellii. And it was associated with a particular condition of infants, where neonates would be born and they'd have these eggs getting past 10 days or 12 days post-birth, which means they were getting infected very, very, very early, which is really unusual. There has to be some unusual transmission pattern for that to occur. And they would get these very swollen bellies, and that was happening because they had a protein-losing enteropathy, and this is ascites, where because of the loss of protein, you're starting to get fluid filling the peritoneal cavity. So these kids were very, very sick. They would end up in respiratory distress, and many of them would die. But the funny thing about swollen belly syndrome was that it kind of happened sporadically. There were two outbreaks in really high up, cold, Central Highland communities in the 70s. And then there were just occasional cases. I know of just one or two occasional sporadic cases since. Another point about these babies is they'd have very, very heavy infection with what look like hookworm eggs, but are actually eggs of Strongyloides, and they're morphologically distinct, but you have to be very good at parasite morphology to tell them apart. So this was a mystery, and it was assumed that Strongyloides philoborni kellii was somehow associated with this, but it may not have been the causative agent, because lots of people were passing Strongyloides eggs, but only a very small number of infants seemed to get swollen belly syndrome. Now, there was one very short read of a region of HVR1 correction of the hypervariable region 1 of 18S ribosomal RNA, sequenced in the early days in 2002. And this was from a sample that had come from a swollen belly syndrome child. And it sequenced as being closely related or closer related to Strongyloides ransomii of pigs and venezuelensis of rats than to stercoralis of humans, which was unexpected and also quite distant from Strongyloides philoborni itself, which is normally a parasite of non-human primates in Asia and Africa. But it's starting to become clear, and our work in Gabon showed this as well, that Philoborni is actually quite common in humans where they're living in environments close to non-human primates. So this was a bit of a mystery. And the funny thing was that this led to it being called Kellii, and some of it just didn't make sense. Anyway, we went back to revisit this, and we got samples that had been used in a microbiome study of babies in Papua New Guinea. There were multiple samples taken over months, and you can see in the red and green dots those which were qPCR positive for Strongyloides. Now, we performed metabarcoding on these at the HVR1, HVR4, and COX1 loci, and this is what we found. So you'll have a look at the phylogenetic tree on the right. We have all these people who have what appears to be Strongyloides philoborni, or all these babies who have Strongyloides philoborni. And this is just the Asian clade of philoborni, identical to the one that you see in monkeys in Asia and in sometimes humans in Asia. And the thing about it is that it's not meant to be in Papua New Guinea. We didn't know that philoborni was in Papua New Guinea. And that's why the epidemiology has been so confused, because there are, in fact, 2 egg-producing species of Strongyloides in Papua New Guinea. But because there aren't monkeys widespread in Papua New Guinea, we assumed that it couldn't be philoborni as the same as the Asian clade, but it actually is. It's just human-to-human transmission of Strongyloides philoborni, which is quite widespread in some parts of Papua New Guinea. Now, what about that other one, that one that was close to Strongyloides ransomii? Well, we didn't get any COX1 sequences on that, but we did get a number of HVR1 sequences, which align perfectly with that one sequence that Doris published in 2002 of a novel Strongyloides species closely related to Strongyloides ransomii of pigs. So that was strange, and we thought, well, that's odd. When I was sort of looking at the draft of the paper, I sort of thought, well, what does ransomii actually do? So I started looking into ransomii. Ransomii is a Strongyloides of pigs, which is transmitted via the transmammary route from the mother to newborn piglets. And those newborn piglets end up with a protein-losing enteropathy, which results in them getting a swollen belly and respiratory distress and ultimately fatal outcomes, which sounds awfully like swollen belly syndrome in Papua New Guinea. In fact, if you look into the history of other zoonotic parasites from pigs in PNG, there have been outbreaks of Ballantioides coli from pigs, particularly in these high central highlands communities, really high up where it's really cold, and they bring the pigs into the hut at night because it's so cold the pigs might otherwise die, so they're in very close contact with the pigs. And we've associated that with quite high rates of a protozoan called Ballantioides coli infecting humans from the pigs. It's interesting that the outbreaks of swollen belly syndrome happened in identical environments to that in Papua New Guinea. And then the occasional sporadic case might just be occasional zoonotic transfer from pigs to humans. So we suspect that pigs are the zoonotic reservoir of this new Strongyloides ransomii-like species that is quite probably the cause of swollen belly syndrome in infants in PNG. So we hope that we can find funding to further investigate that, because it's actually very, very important, and it's a new Strongyloides of humans that may be far more widespread and far more clinically important than we realise. So this is just a bit of a brief summary of some of the things which I've been doing in my research. I wanted to quote William Shakespeare, "There are more things in heaven and earth, Horatio, than are dreamt of in your philosophy." And I like to say there are more parasites in heaven and earth than are dreamt of in your philosophy. And as we go back and employ these sequencing approaches to what have previously been well established and common, and we think we know everything about them, nematodes of humans, we're finding that actually we're discovering a whole lot of new pathogens, and all of them have implications for the pathways of transmission and for the WHO Global Control Program for soil-transmitted helminths. So there's a lot to still be discovered in parasitology, and it's really an exciting area once you start looking into using sequencing to discover new pathogens, combined with, I might add, traditional microscopic morphology, which there is still a place for, and I always talk about people, we need to maintain those parasite microscopy skills. So thank you very much for listening to my talk. I hope you found it interesting and that you've learned some new things about nematodes. If you have any questions, please feel free to email me. I'm really interested in talking about any of this. So thank you very much for your time, and I hope you've enjoyed the talk. Thank you.